Some viruses insert their genome into our genome, and if you don't know what a genome is, it's their genetic information. They put their DNA into our DNA, and once they do this, we can never get this DNA out of our cells again. It becomes a part of our DNA. These little guys are called retroviruses, and the bits of DNA they leave behind in our DNA are called human endogenous retroviruses, or HERV for short. Welcome, ladies and gentlemen. I'm Nick, the veterinary guy, and today on Science Scoops, we're tackling one of my favorite topics in current research, all about placenta and all about viruses. HERVs make up a shocking 8% of our genome, and these sequences are often termed junk DNA, but I'm here to tell you today that we're discovering that some of these endogenous retroviruses are turning out to be way more important than we could have ever imagined. During pregnancy, the placenta delivers oxygen and nutrients from the mother's side to the baby, but it's really important during this process that the mother's blood and the baby's blood do not come into direct contact. For this reason, there are special barriers in the placenta that are separating these two so they don't actually get in touch with each other. Researchers studying the placenta during the 90s noticed a surprising amount of virus-like particles in it. So when they sequenced the placental DNA, they found a human endogenous retrovirus, a HERV, that has acquired a really important task during pregnancy. This particular HERV gene, called syncytin, produces a protein also called syncytin. And this protein is known to fuse together several cells into one big layer with several nuclei. Now, viruses actually use this syncytin protein to create their envelope, which is kind of a sticky protective layer that they have outside of their capsule. But what on earth is this gene doing in the human placenta? Well, it's exactly in the placenta where this ability to create a specialized multinucleated protective layer that functions as a barrier is incredibly important. This green layer is the one we're talking about right now. This originates as a collection of cells and is then fused together by the syncytin protein, which is produced by the syncytin gene, which came from a virus. Are you sticking with me? This syncytin gene has proven to be so essential during pregnancy that when an experiment was done on lab mice where they knocked out the gene, this means that they inactivate it, so it's not producing any of that protein, and they don't change anything else. So only this gene is inactivated, and in the embryos, mouse embryos, these embryos were actually not able to come to term. They would repeatedly pass away in the uterus. Now, obviously, we cannot perform this kind of experiment in humans, but these mammalian syncytin genes, originally viral syncytin genes, have been found so far in every mammal that has been researched for it, and the consensus is really clear that this is an essential gene for the formation of the placenta and so for the successful pregnancy. So what does this all mean? Somehow, through the miracle of evolution and horizontal gene transfer, this viral gene that we received millions of years ago plays an essential role in us being able to carry a full pregnancy to term. But what's even more interesting is that in many different mammal species, different types of syncytin genes have been found. And we've discovered that these syncytin genes are not even related to each other, which means that this whole scenario where a mammal received a viral gene and then it turned out to be the essential puzzle piece missing to start developing a placenta did not happen just once, but actually countless times again and again in different mammal species. And this also might explain why the placenta is one of the most variable organs in the class of mammals, since an essential gene came from all types of different viruses at different times, leading them to develop completely differently from each other. If I showed you a cow placenta, you might not even recognize it because it's so different from a human one. Now, since mammals are defined as a class by having a placenta, with two exceptions, this only begs one last question. Which came first, the chicken or the egg? Or in this case, the mammal or the viral syncytin gene? Thank you so much for watching today's video. I thoroughly hope you enjoyed it and perhaps even learned some of it. Make sure to check out these other videos I've picked for you that I hope you will enjoy an equal amount or even more. And of course, subscribe to this channel if you want to be up to date with fun science, and also maybe if you're studying veterinary medicine, because I love to talk about that stuff.